of years ago look at iPS cells and found hotspots, so-called hotspots of variation, where no matter which lab derived the iPS cells, whether they're from San Diego or from Boston or from Madison, Wisconsin, through our collaborators, we could always find there were some regions that were problematic, that they had either a memory of their parental cell, of their skin cell, or they have regions that are common to all iPS cells. And one thing we could find actually in this study is that the iPS technology is improved, because we can see that the cells that were made as part of this study actually they have less problems than the one even of 2 years ago. So the culturing conditions, growing them in low oxygen, etc., has actually helped, I think, to improve iPS technology. And I think that's what we're sort of experimentalists, not clinicians, thinking that we can get out of this, is the understanding of what is different, what is missing, that you could add to iPS cells to make them look more like ES cells, could be derived from an understanding of what the egg cytoplasm is actually providing in terms of the reprogramming.
Yeah, that's what I was just going to ask about. There's something inside there that seems to help a more faithful method of reprogramming.
Yes.
Okay, so if you can just find out what it is, and I imagine you're probably working on that.
Yeah, well, in collaboration with Shokrat's lab, there's a number of other experiments that he's, lines of like looking at, for example, the age.
Yeah.
So these were cells from actually fibroblasts that were fetal fibroblasts, and now you can look. In fact, there was just a nice publication by another group from New York in Nature. That describes looking at actually some individuals that are adult and also diabetic, where the SCNT cells were made, Dieter Egley's group, and so looking at how well those nuclei get reprogrammed will also be important, or even among different people. Again, this was a well-controlled experiment with only one genetic donor, essentially, and so now you'd want to look at at the different ages of the cells, different types of cells. Maybe they're equally reprogrammed as well, and maybe they're not by the egg cytoplasm. So I think there's a lot of work still to do. But I think the good news is that right now there are cells that potentially are maybe a better route for therapeutic treatments.
All right. And anything else you want to say about the significance of the paper and possible follow-ups?
Yeah, I think it was the next obvious thing to do. In fact, even in the publication by Egley Group, the very last paragraph describes, we should probably be looking at the epigenomes of these now that they were able to derive them. And so it was sort of the next logical thing to do, is to ask how close are they to ES cells by very high resolution sequencing methods. And so now there's a whole variety of things, as I mentioned, that could be done to compare different ages of cells and different quality of eggs, for example. Not all eggs were created equally, it turns out, in terms of looking at efficiency of reprogramming. So there's still a lot to do, but I think it looks very promising.
Okay.